Assalamualaikum warahmatullahi wabarakatuh. I'm Ahmad Afiq Aiman bin Amran, matrix number S58692. My supervisor is Professor Madya Dr. Siti Nordahliawati binti Muhammad Siddiq. Today, I would like to present my final year project entitled Soil-Borne Fungal Pathogen at Durian Orchard. Problem statement. Stem canker caused by the fungal pathogen called Phytophthora palmivora poses a significant threat to durian orchards, leading to economic losses and reduced fruit quality. Then a significant challenge in maintaining optimal soil nutrient levels and the availability of these nutrients in plant tissues to support pathogen growth and development, and reviews the effects of the different nutrients on disease development. My objective through this project are firstly to identify fungi that reside in the soil at different durian orchard, and secondly to determine soil favorable conditions, which is soil particles and elements by fungi at different durian orchard. So my sampling location are located at Melaka, which is at Jasin and Merlimau, and Johor, which is at Muar and Pagoh. For methods, I use soil sampling and durian stem canker sampling. After that, I do the pathogen isolation and obtain a pure culture, and then identify the fungi morphological characteristics. Next, I do the soil analysis to obtain the soil nutrients, particles, and soil pH. First, stem canker sampling. The stem of durian tree was cut out with sterile knife after symptoms was identified, which is brown to reddish lesions on the stem. Then sample kept in zip bag. For soil sampling, the composite soil was collected for 10 samples for each orchard. The soil are collected at different orchard, so total 40 sample of composite soil. For one sample, a hole approximately 2-inch depth was dug and the sample took with a sterile scoop at different points underneath the durian canopy. The composite soil kept in a container to keep moisture. Methods I use to isolate the pathogen is soil baiting. The soil baits I use are green apple and durian leaf as a host. Firstly, 60 grams of composite soil was weighed and then flooded with 60 ml of sterile water. Let it incubated for 2 until 4 days. After that, the apple was cut into slices and embedded into soil and then left under room temperature for 2 until 4 days. Then, the part of embedded apple was cut, let dry and put into CMA plate for fungi growth. Next is for soil baiting for durian leaf. The procedure are same, which fresh durian leaf were cleaned and sterilized with 70% ethanol. Then it was cut into square about 1 cm x 1 cm and let it dry. The leaf were left floated above soil water in room temperature. After 2 to 4 days, the leaf transferred to CMA plate. After the fungi grow about 2 or 3 days, the fungi can be isolated from CMA plate or leaf host to PDA plate. Then, to obtain a pure culture, next isolation I done, which transfer it into new PDA plate, which is free from any contamination by other microorganisms or single fungi only. Next I do is microscopic observation from a pure culture to observe the morphological of particular fungi. So for example, this is a morphology for Fusarium species that I obtained under a microscope. Soil analysis I have done are soil pH, soil texture using particle size analysis, and then soil nutrient using CHNS analyzer and ICPOS machine. So this is summary of data collected. First is for soy baiting. Soy baiting that using apple bait resulted fusarium is higher species found compared with durian leaf. Colepotrichum is higher were obtained. Oomycetes, which is I not classified the specific species yet, found higher in durian leaf baiting compared with apple bait. There are also less contamination with bacteria and saprophytes in durian leaf soy baiting compared to apple bait. Biocontrol such as Penicillium, Trichoderma, and Aspergillus also found in both baiting. For soil analysis, particle size analyzer is used to identify the soil texture of composite soil. From this graph, we found that soil orchard at Muar, Johor, Yasin and Merlimau, Melaka has a silt soil, which is 2 to 50 micrometer. Meanwhile, Pagoh, Johor has a sand soil, which is 50 to 2000 micrometer. Then soil analysis using CHNS analyzer for soil nutrient observation, which are carbon, hydrogen, nitrogen, and sulfur. We found that overall orchard have higher percentage of carbon, and Muar Johor is the highest. Hydrogen is second element and nitrogen is the lowest, and sulfur is 0 percentage for each orchard. Next is soil pH. Muar range pH 4 to 6 and Jasin 5 to 6. Meanwhile, Pagoh and Merlimau have pH around 4 to 5, which is the most acidic soil pH found. There are also problems while conducting in this project. For example, many plate contamination using apple baits, especially bacteria due to not properly dried, while transferred to agar plate. So I repeat the soil baiting method using different bait, which is durian leaf as a host for fungi. I also apply proper procedure to avoid more contamination. Second problem is the machine was broke down when I want to analyze the nutrients, which is ICP-OS machine. So I keep updating with the stuff until the machine is well functioning. Luckily this month the ICP-OS machine is functioning and I can run my samples. I'm sorry for not inserting the data for ICP-OS yet for this presentation. So this is my Gantt chart and my project progress is 70%. I'm doing data analysis and then this waiting. These are my references. That's all from me. Thank you.